Investigadores de dos universidades estadounidenses anunciaron que encontraron una abundante fuente de las llamadas células troncales o células madres en el líquido amniótico, el que protege a los fetos en la matriz. Agregaron que produjeron con ellas una variedad de tipos de tejido que podrían emplearse en reparar órganos dañados.
While pregnant, often that liquid is where the liquid that's withdrawn from the woman's belly is what you found these stem cells.
So when the baby's born comes the afterbirth, which is the placenta, and we found the same stem cell population there as well.
And how much would you say in a pint of this fluid? How much of this fluid contains stem cells, viable stem cells?
Actually, the fluid is chock full of cells because the embryo is constantly shedding cells. But we were looking for a stem cell population, a cell that we could drive to become other things, that would be nimble. And that particular cell makes up about 1% of the cells in the fluid and the placenta.
Describe what you mean when you say a cell that could be nimble.
Well, basically, it's a cell that has the ability to become other tissues. And therefore, we have been able to drive the cell to what we called all three germ layers, which basically means all three major classes of tissues available in the body from which all cells come from.
And that's bone, muscle, what?
Well, that involves basically all tissues in the body, but we actually were driving it to specific tissue types such as nerves, bone, muscle, cartilage, the cells that line blood vessels, liver, and other tissues like that.
Now, there's been a discussion or a debate in this country about stem cell research for some time. This is different. This kind of amniotic stem cells that you're talking about is different from, say, adult stem cells. How?
That's right. It's actually different from both adult and human embryonic stem cells. It has properties of both. It differs from adult stem cells in that they can grow very rapidly. So in that way, they act more like human embryonic stem cells. They double in number every 36 hours, just like human embryonic stem cells.
So if there were a continuum with embryonic stem cells coming from the embryo on one end and adult stem cells come on the other end, this falls somewhere in the middle in terms of its ability to replicate?
It's actually much closer to the human embryonic stem cells. In fact, it has almost identical replicating abilities as the human embryonic stem cell. So it's one of the advantages of the cells is that you can grow these in very large quantities for therapy.
So you talk about the advantages. What are the advantages? Who would benefit from the use of these cells if they were to turn out to be as fruitful as they seem, and how would they benefit?
Well, I think that patients may benefit from these technologies, but that's still years down the line. These cells are still early in their development, and like human embryonic stem cells, we are still trying to figure out how to get those cells to therapy. The same thing will be true for these cells as well.
So what do you do with these stem cells? You put them in a freezer and hope that one day they're useful?
Well, actually, there are two ways to get these cells for use. One of them is to preserve the cells at the time of birth and freeze those cells down so you can have a natural repository of cells available to that baby throughout their lifetime in case they need it for a medical condition. Or an alternate way to do this is to actually bank these cells. And you need approximately 100,000 specimens in a bank to provide about 99% of the U.S. population with a perfect genetic match for transplantation.
Once again, some years down the line, in theory, right?
That's right. I think at this point we are driving these cells to become nerves and bone and cartilage and muscle. But these are still experimental studies, and it's going to take several years before we can get this to the patient.
What are the potential medical applications if everything were as you would hope, then these were as useful as they seem? What would be the medical application?
Well, for example, in this study we described how these cells can be converted to nerves that can then be injected into areas of brain injury, and they're able to replenish the brain injury site in experimental models. So the hope for that, of course, would be for patients with Parkinson's or stroke disease or spinal cord injuries. But again, this is still very early in its development.
Has this been tested in living beings yet, like, say, in mice?
This has been tested in mice. So actually, nerves have been injected into mice where they replenish the brain-injured area. They have also been injected into mice to replace bone. We have been able to make liver cells that secrete urea, which is an important component of what the liver does.
And there have also been other experiments done which have been done in sheep, I gather.
That's right. So these cells actually have been in the amniotic fluid for many years, and we've known about their presence for many years. The major finding in this study is the fact that we found a stem cell population that is a true stem cell in terms of their nimble ability to become old tissue types.
So does this end the argument that we've been having all these years about stem cell research?
It does not, mainly because it's another stem cell choice, and I think you really can't tell which cell is going to be best for which indication, and all cells have advantages and disadvantages.
Dr. Anthony Atala, thank you very much for helping us with that.
Nice to be with you.